morning, viewer. Welcome to our Glory Globe TV. Here's the breaking news. Yes, which types of cells can be located in various human tissues and where? Which genes show activity in these individual cells and which proteins can be identified within them? Detailed answer to these inquiries and more are expected to be supplied by a specialized atlas. This atlas will particularly elucidate how different tissues take shape during embryonic development and the underlying causes of disease. In the process of developing this atlas, the researchers have the goal to chart not just tissues directly procured from humans but also structures referred to as organoids. These are three-dimensional tissues aggregates that are grown in the lab and developed in a manner similar to human organs, albeit on a similar and smaller scale. The advantage of organoids is that we can intervene in their development and test active substances on them, which allows us to learn more about healthy tissue as well as disease, explains Barbara Talent, Professor of Quantitative Development Biology at the Department of Biosystem Science and Engineering at ETH Zurich in Basel. To help produce such an atlas, Talent, together with researchers from the universities of Zurich and Basel, has now developed an approach to gather and compile a great deal of information about organoids and their development. The research team applied this approach to these organoids of the human retina, which they derived from stem cell. At the heart of the methods the scientists used for their approach was the 4I technology, iterative indirect immunofluorescence imaging. This new imaging technique can visualize several dozen proteins in a thin tissue sections at high resolutions using fluorescence microscopy. The 4I technology was developed a few years ago by Lucas Pelkmans, a professor at the University of Zurich and co-author of the study that has just been published in the scientific journal Nature Biotechnology. It is in this study that the research applied this method to organoids for the first time. Behind the report, Glory Globe TV, Delhi, India.